Every second, trillions of cells in your body are working to grow, repair tissues, and maintain the complex systems that keep you alive. This incredible feat is made possible by the cell cycle, a carefully regulated process that allows cells to divide and create new cells. However, when this cycle is disrupted, the consequences can be profound, leading to the development of diseases such as cancer. Today we'll explore how disruptions in the cell cycle occur, why they happen, and how these changes can result in disease. To understand how the cell cycle can go wrong, we first need to understand how it normally works. The cell cycle consists of two main phases: interphase, where the cell grows and prepares for division, and the mitotic phase, where the cell divides into two identical daughter cells. Interphase is further divided into three stages: G1, GAP1, S, synthesis, and G2, GAP2. In G1, the cell grows and performs its normal functions while preparing for DNA replication. During the S phase, the cell duplicates its DNA, ensuring that each new cell will have a complete set of genetic instructions. G2 is the final checkpoint where the cell checks for errors in DNA replication and prepares for mitosis. The mitotic phase then completes the cycle, with the cell dividing into two new cells. Throughout the cycle, a system of molecular checkpoints ensures that everything proceeds correctly. These checkpoints monitor the cell's size, DNA integrity, and readiness for division. If something goes wrong, like DNA damage or incomplete replication, these checkpoints can halt the cycle and allow the cell to repair itself. If the damage is irreparable, the cell may self-destruct through a process called apoptosis, or programmed cell death, to prevent further harm. But what happens when these checkpoints fail? This is where problems arise. Disruptions in the cell cycle can lead to uncontrolled cell division, a hallmark of cancer. Cancer is essentially a disease of the cell cycle, a condition in which cells divide uncontrollably and invade surrounding tissues. The root cause of these disruptions often lies in the cell's DNA. Mutations, or changes in the DNA sequence, can alter the function of genes that regulate the cell cycle. Two types of genes play a critical role in this regulation: proto-oncogenes and tumor suppressor genes. Proto-oncogenes are like the gas pedal of a car. They promote cell division when needed. When these genes are mutated, they can become oncogenes, which cause the cell to divide uncontrollably, even when it shouldn't. On the other hand, tumor suppressor genes act like the brakes. They slow down or stop the cell cycle when necessary, such as when DNA damage needs repair. If a tumor suppressor gene, like the well-known p53 gene, is mutated or deactivated, the cell loses its ability to halt division. Without these brakes, damaged or abnormal cells can multiply unchecked. Environmental factors can also contribute to cell cycle disruptions. Exposure to carcinogens, substances that cause cancer, can damage DNA and lead to mutations, and certain chemicals. Viruses can also play a role in disrupting the cell cycle. For instance, the human papillomavirus, or HPV, produces proteins that inactivate tumor suppressor genes, increasing the risk of cervical and other cancers. Once the cell cycle is disrupted, abnormal cells can begin to accumulate. These cells may form a tumor, a mass of tissue that arises from uncontrolled cell growth. Tumors can be benign, meaning they don't invade nearby tissues, or malignant, meaning they spread and damage surrounding structures. Malignant tumors are the hallmark of cancer and can metastasize or spread to other parts of the body through the bloodstream or lymphatic system. As cancer cells spread, they disrupt the normal functions of tissues and organs. For example, cancer in the lungs can interfere with oxygen exchange, while cancer in the liver can impair detoxification processes. This systemic impact is why cancer is such a devastating disease. Understanding the link between cell cycle disruptions and cancer has led to important advances in prevention, diagnosis, and treatment. For example, early detection of mutations in genes like BRCA1 and BRCA2, which are linked to breast and ovarian cancers, allows individuals to take preventive measures. Treatment strategies often target the uncontrolled cell division that characterizes cancer. Chemotherapy and radiation therapy, for instance, aim to kill rapidly dividing cells. More recently, targeted therapies have been developed to attack specific molecules involved in the cell cycle, offering a more precise approach with fewer side effects. One of the most exciting developments in cancer treatment is immunotherapy, which harnesses the body's own immune system to recognize and destroy cancer cells. By targeting proteins that allow cancer cells to evade immune detection, these therapies can restore the HPV vaccine protect against virus-induced cancers, highlighting the importance of public health measures in disease prevention. In conclusion, the cell cycle is a finely tuned process that ensures the proper growth and division of cells. When this process is disrupted, whether by genetic mutations, environmental factors, or viral infections, it can lead to uncontrolled cell growth and the development of diseases like cancer. Understanding these disruptions not only deepens our knowledge of how cells work, but also paves the way for innovative treatments and prevention strategies. By continuing to study the cell cycle and its regulation, scientists are bringing us closer to a future where cancer and other cell cycle-related diseases are no longer life-threatening.